The overall goal of the following experiment is to examine the telomere G-overhang structure in Trypanosoma brucei cells. Native in-gel hybridization uses a sequence-specific oligoprobe to hybridize to the single-stranded region of the telomere under the native condition. This single-stranded region is the telomere G-overhang region, and therefore the amount of hybridized oligoprobe represents its length. Adapter ligation allows the ending sequence of the telomere G-overhang to be determined when the unique oligo is labeled. This method involves the ligation of a sequence-specific adapter to the end of the telomere G-overhang structure. For the ligation-mediated primer extension assay, the guide oligo is end-labeled and annealed to the unique oligo before it is ligated to the chromosome ends. After ligation, primer extension is performed using T4 DNA polymerase. The length of the G-overhang can then be determined, as the product is N-6 nucleotides longer than the actual G-overhang, where N represents the length of the guide oligo in the adapter.
Hi, I am Bibo, an assistant professor in the Department of Biological, Geological, and Environmental Sciences at Cleveland State University.
Hey, I am Ranjodh Sandhu. I am a graduate student in Dr. Lee's lab, and today we are going to show you a procedure to analyze G-overhang structure.
We use this procedure to study telomere structure and functions in Trypanosoma brucei. When working with radioactivity, as in this experiment, it is necessary to take proper safety precautions. Institutional environmental health and safety department should be consulted before this is attempted. In this video, some of these precautions are absent for demonstration purpose only.
So let's get started.